My thesis topic is evaluation of Sabella spallanzanii and Microcosmus squamiger collected in Southern Portugal as a source of bioactive ingredients for the aquaculture industry. The study is under the supervision of Dr. Luisa Custodio and Dr. Sofia Ingrola from the Center of Marine Science, University of Algarve. According to the information from the Food and Agriculture Organization, aquaculture represents more than half of global fish production for human consumption. From 2001 and 2018, the annual growth rate of aquaculture was 5.3%. It is inevitable that aquaculture is advancing in food production. However, this does not fully ease the pressure on wild stocks, since it still calls the ecosystems by the need of exploited low commercial fishery to feed the aquatic products. Fishmeal is made from pelagic fish species that are suitable for human consumption. Despite the fact that with the effort of the researchers, the aquatic industry has decreased the inclusion level of fishmeal, it is important to explore normal feed sources from a lower trophic level and outside the human food chain. To increase the sustainability and reduce the dependency of fishmeal, numerous studies have focused on identification of alternative feedstuff. Due to low cost and high accessibility, plant-based feeds such as soybean meal are used as primary fish meal alternative, but the presence of anti-nutritional compounds, along with the lack of essential amino acids and low palatability, make it not the ultimate alternative of fish meal. Apart from the nutritional value, antioxidant properties are also important in fish feeds. Antioxidants stabilize the lipids present in the feed. Oxidation will compromise the quality of aquatic products. Antioxidants has been introduced into the feeds to prevent oxidation. However, the adverse effect of synthetic antioxidants remains inconclusive. For example, axoziquine has been found to pose negative impacts on immune response and resulting abnormality of organs in fish and shrimp. So the potential hazards of synthetic antioxidants encourage researchers to explore natural antioxidants. Sabella spallanzanii, also known as Mediterranean fangworm, a native species from the Mediterranean Sea and southern European Atlantic coast, has recently invaded Australia and New Zealand coastal water. It is commonly found on the sediment substrate or artificial hard surfaces to a depth of 30 meters. Sabella spallanzanii has already been reported as a bioremediator of aquaculture waste and could be integrated into multi-trophic aquaculture system. Another marine vertebrate, Microcosmus squamiger, is also an ideal candidate. It is a solitary ascidian native to Australia and documented as an invasive species in Portugal. It is now considered as a cosmopolitan species. Microcosmus squamiger inhabits hard littoral substrate in a depth of 10 meters. The closely related species, Microcosmus experitus, has been reported high protein content. Make Microcosmus squamiger also worth a check. To the best of my knowledge, there is no evaluation of samples from the Portuguese coast, and nothing is known about the antioxidant properties of these two species. The end of this study was to evaluate the bioactive components of Sabella spallanzanii and Microcosmus squamiger and their potential use in the aquaculture feed industry. In this context, the proximate composition, amino acid content, minerals content, antioxidant activity of extract of these two species were analyzed. Adult animals were hand collected as bulk together from the floating dock in Faro in January 2020. The moisture were that measuring by drying the biomass at 60 degrees Celsius, while the ash content were determined by incineration of the samples at 525 degrees Celsius. After the biomass was freeze-dried, the biomass was sent to test the protein content using macrocudal method 25, and total lipid were determined by extracting the freeze-dried biomass in accordance with chloroform and methanols. Then the gross energy will calculate by hot water general factor systems with protein content and total lipid. The amino acid profile was performed by ultra-high performance liquid chromatography, and mineral content were analyzed by microwave plasma atomic emission spectroscopy after freeze-dried biomass digesting with nitric acid and hydrogen peroxide. Unfortunately, the fatty acid analysis was cancelled due to the COVID-19 situation. The biomass after the biomass was collected, the extract were obtained from ultrasound assist extraction with dichloromethane and methanol respectively, in a ratio of 1 gram of freeze-dried biomass and 40 milliliter of the solvent. After we suspend the extract in DMSO, the samples were subject to the test of antioxidant activity. The results were demonstrated in IC50 for DPPH radical scavenging activity, iron chelating activity, and copper chelating activity. The lower the value, the stronger the activity. The oxygen radical absorbance capacity, ORAC, was demonstrated in microgram of Cholox equivalent per gram of extract. The higher the value, the stronger the activity. All results present as mean with standard deviation. One way ANOVA was performed when needed. So for the Proximate composition, both species had high moisture content. The moisture content of marine vertebrate varied. A review of 35 marine invertebrates gave moisture content range from 77 to 92%. The ash content of Sabella, spallanzanii, and Microcosmus squamiger was higher than the previous study. A high ash content does not necessarily bad. Whole blue mussel was used as an ingredient of diet for rainbow trout, though the shell contained about 80% of ash. It is notable that both species have high protein contents when compared among different marine vertebrates such as mussels and Japanese sea cucumber. The high protein content is one of the reasons that make Fishmeal a good feed sources. A nice fish meal could contain up to 72% of protein. The total lipid content of both species are in the range of other marine vertebrates. Most of commercial fish feed contain total lipid from 10 to 25%, act as a source of energy, cell membrane function maintenance, and the precursors of physiological process. The estimation of the gross energy in Sabella, spallanzanii, and Microcosmus squamiger was 12.8 and 11.6 respectively. A total of 21 amino acids were identified. There was a prevalence of arginine in Sabella, spallanzanii, and in Microcosmus squamiger, taurine was identified as the dominant amino acid. Fish cannot synthesize 10 essential amino acids endogenously. Unlike Fishmeal, have a balanced amino acid profile, providing all essential amino acids. Sabella, spallanzanii was found particularly abundant in arginine. Deficiency of arginine in diets result in reduced growth and low feed efficiency in Japanese flounder, milkfish, and Atlantic salmon. In this context, Sabella, spallanzanii could be an ideal source of arginine. In Microcosmus squamiger, taurine was identified as the dominant amino acid. Though it is not an essential amino acid, deficiency of taurine can lead to green liver syndrome and poor growth performance in Japanese amberjack and red sea-bream. It is noteworthy that soybean meals contain a very low concentration of taurine. Although the supplement of synthetic taurine is considered effective and can be added in animal diets in Europe, in US, the use of taurine as additive is not allowed only if it is added indirectly, according to local Food and Drug Administration. In this framework, Microcosmus squamiger can act as a good selective additive of chelating in compliance with the local regulation. The major minerals found were aligned with the most concentrated metal in seawater. Toxic heavy metals were not detected. Because marine fish can absorb minerals directly from the water through gills and skin. So for the following, I will only discuss the minerals which are relatively low in seawater. Phosphorus was found in both species. It is an essential component of fish diets that is crucial for skeletal development and intermediary metabolism. Phosphorus was usually present in an excess amount in formulated fish feed due to the high level of fishmeal and animal byproducts they included, as there was no apparent danger from overprovision. However, the supplement of phosphorus in fish feed is a notable source of eutrophication. Therefore, the supply of phosphorus is a matter of balance between meeting the requirement of fish and controlling environmental impact. Fishes too. Iron is an important micronutrient for teleost fish, known as the integral component of proteins, participates in cellular respiration and oxygen transfer. Iron is required in a diet for Atlantic salmon in a concentration of 60 to 100 mg per kilo. However, an excess amount of iron may cause the disease winter ulcer in Atlantic salmon. A trace manganese was found in Microcosmus squamiger. Supplementation of manganese is important since it acts as cofactor for several enzymes. Lack of manganese could lead to dwarfism in fish. The requirement of manganese in fish range from 2 to 20 mg per kilo, depending on the fish species. For the antioxidant activity, BHT and EDTA act as positive control here. Methanol tend to have a higher extraction rate than dichloromethane. Despite there is no activity towards DPPH in our test, as you can see, the methanol extract of Sabella spallanzanii demonstrate the capability of chelating iron and copper. Also, the methanol extract of Microcosmus squamiger chelate copper. Both iron and copper are essential elements for fish metabolism. However, excess amount of metals are inducer of oxidative stress, promoting the formation of reactive oxygen species, ROS. ROS can damage proteins and lipids, which is deleterious to fish health status. Study had reported the benefit of dietary EDTA supplementation in Nile tilapia, which enhance the antioxidant activity and reduce heavy metal bioaccumulation. So both species can be a potential source of natural chelator, protect farm fish from heavy metal exposure. Depending on the species and the extracting solvent, ORAC value varied from 14 to 26.3 microgram Cholox equivalent per gram of extract. But all ORAC values observed here are considered low when compared to strong natural extract from plants such as the grape skin and the grape seed. The value from Atlantic sea cucumber is also quite low. However, the values are in the range of fresh fruits and vegetables. In this work, the high protein content with specialized and no toxic heavy metal detected suggests the species as a potential normal feedstuff. The metal chelating characteristic could protect the fish from oxidative stress induced by heavy metal exposure. This is the first time to quantitatively evaluate the antioxidant property of Sabella spallanzanii and Microcosmus squamiger collected from the southern coast of Portugal. Further research is needed to evaluate the palatability and incorporation potential of Sabella spallanzanii and Microcosmus squamiger in diets for fish species. I would like to thank my promoters, also experts who assist me. Without their helpful input, this research could not be could not be conducted, especially under this unique global crisis of COVID-19. Also, thanks for the endorsement from my family and everyone I have met through my IMBRSea journey. Here comes the reference.